Can somebody please confirm you can hear me?
We can hear you.
Great. I'm gonna shut that down. All right, before we begin, are there any questions? Today is February 11th, and we are supposed to be discussing Chapter 7, Throat and Urine Cultures. I will send you the unknown results for the first 3 tests sometime after this lab. So something like 7:30 to 8:30 is my goal for sending it out. Any questions about anything? Let me remind you, I mentioned in the lecture that you have a quiz 3, tomorrow, next Wednesday, this coming Wednesday. All right. Let's go to the lecture. Lab 7, throat and urine cultures. There is some reading to do in this for this lab, but it's only a page about nose, throat, and mouth, normal microbiota. There's some reading from the appendix at the end of this lab module about beta hemolytic streptococci. There's also some information about Haemophilus at the in the appendix of this lab module. And also, I guess I'll put that there. It's out of place. But coagulase, I've got a little paragraph or two about coagulase at the in the appendix to this lab module. And then a page 132 on oxidase test and a page on the catalase test, page 155. So not much reading for this lab. Any questions about any of that? We do have some video clips in this lab. Your learning objectives for this lab are: 1, be able to explain how to properly collect throat and urine specimens for routine culture in the lab. 2, list common bacterial pathogens isolated from throat and urine cultures. 3, know the culture media used to cultivate these pathogens. 4, recognize which tests are used to identify throat and urine pathogens. And 6, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. So we'll talk about these tests and you should understand them and be able to explain them. We will briefly talk about the XV test, but you don't need to understand it or explain it. Meaning I'm not going to test you on the XV factor testing. Any questions about these learning objectives? All right. Once specimens are received in the lab, they are logged and processed in a timely fashion with specific culture media inoculated and incubated for throat and urine specimens. Below is a table of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For the throat, we only have 2 culture media that we use. Most of the time, we'll be culturing the throat cultures on blood agar. Some of the time, especially in the case of children, small children, and infants, we will also use chocolate agar. Streptococcus pyogenes is the main pathogen obtained from throat cultures. Streptococcus pyogenes, if you do not know, is the bacteria species that causes strep throat. However, in children, especially small children and infants, You may also culture Haemophilus influenzae, which is a pathogen in small children and infants. And Haemophilus influenzae will not grow on blood agar. It can grow on chocolate agar. Streptococcus pyogenes grows on blood agar. It does also grow on chocolate agar. You don't need to know that, but it does. All right, that's it for the throat specimens. How about from the urine specimens? We have two media that we always use for a urine specimen. So we always use blood agar, and then we use either McConkey's agar or EMB using methylene blue agar. So we always use two, blood agar and one of the others, and then the other is either McConkey's agar or EMB agar. Blood agar grows everything. It will also grow fastidious organisms, or at least some fastidious organisms. McConkey's agar and EMB agar only grow gram negatives. We use blood agar and McConkey's or EMB because most of the pathogens obtained from the urine are gram negatives, and that's why we use McConkey's agar or EMB. But some of the pathogens are gram positives, and to get them, you have to grow it on blood agar or some other media. So some of the pathogens we find from urine cultures, like a urinary tract infection, is Streptococcus, Enterococcus, and Staphylococcus, and all three of these are gram positives. But we also have pathogens of family Enterobacteriaceae, particularly E. coli, and then the Pseudomonas family. And these two families are gram negatives. The number one species causing a urinary tract infection is E. coli. All right, for throat cultures, a sore throat can be from a virus or a bacteria. If it is from a bacterial infection, we'll often see white patches or spots on the back of the throat or the tonsils. And this one shows a little bit of white, but a little less intense white on the tongue as well. For a viral cultures, we do not see the white patches or spots, and it'll just be a red throat. So with bacteria, you have red and white patches. And with virus, it's just red. The most commonly isolated bacterial pathogen from the throat is Streptococcus pyogenes, and that's the name I use, the name of the species. But it's also got an older name, Group A Streptococcus, and that's relying on the antigens for the Streptococcus family. And I guess they used that system earlier when they didn't know the species. And group A streptococcus is sometimes abbreviated GAS, or the G for group, A for A, and then S for streptococcus. Any questions about any of that? Okay, Streptococcus pyogenes causes strep throat. How you test for strep throat is you swab the back of the throat or the tonsils or both where there's redness or white patches. You avoid touching the tongue, the sides of the mouth, and the top of the mouth. A tongue depressor tends to help. Patients that test positive for Streptococcus pyogenes need to be placed on antibiotic therapy. The reason is that Streptococcus pyogenes has a capsule around it, making it very difficult for the patient to remove and attack the bacteria. Streptococcus pyogenes, or group A strep, is a fastidious organism, and it must be cultured on a plate with blood in it, usually 5% sheep's blood, and that's called the blood agar plate or BAP. And so we just swab their throat or tonsils or both, and then swab the plate with the swab. And then you grow the plate and look for small colonies, usually white colonies, surrounded by a complete zone of hemolysis, which, if it's having beta hemolysis, it could be Streptococcus pyogenes, but there are other members of the Streptococcus family which could be giving the group A strep. And then, of course, there's Staphylococcus aureus, which is also capable of giving beta hemolysis. There's a few other bacteria that can give beta hemolysis. But once you got beta hemolysis on the blood agar, there are only two biochemical tests required for definitive identification of Streptococcus pyogenes. And that's using the catalase test and the basis tracing susceptibility test. So here we have a blood agar plate streaked from somebody's throat. And you'll notice that this region of the plate is actually, let me blow that up, alpha hemolysis. That one there, excuse me, not alpha hemolysis, gamma hemolysis. This one there is definitely alpha hemolysis. This darkening of the plate in this region of the plate is also alpha hemolysis. The point is there's no beta hemolysis. On this one, we have clear beta hemolysis, where looking at this bacteria here, in the very center, you see the white area. That's a very small colony of Streptococcus pyogenes, which usually gives only small colonies. And then there's a clear region around the bacteria, and that's from beta hemolysis. We can see lots of beta hemolytic activity around all of these colonies, and then around this smear there, and then there, and there. I'm not sure why it's happening in these three places there and not so much in the rest of the place. It's probably happening very small increments there, but it's not forming a colony because there isn't a colony. There's too much growth. But for some reason, you're seeing large amounts there. I don't know why. All right, once you get beta hemolysis on a blood agar from a throat culture, you then need to perform two tests to show that it is Streptococcus pyogenes. The first test is the catalase test. It's looking at the enzyme catalase. Catalase, in the presence of hydrogen peroxide, will make the hydrogen peroxide convert to water and oxygen. The oxygen will create bubbles. And so we look for the production of bubbles. And if you see bubbles, it's catalase positive. If there's no bubbles, it's catalase negative. Any question about any of that? Most bacteria that grow in the air have the enzyme catalase, and so they will be catalase positive. However, members of the Streptococcus family and the Enterococcus family grow in the air, but they are catalase negative. You then do the bacitracin test. Bacitracin is an antibiotic. So what you do is you take the bacteria obtained from the blood agar plate and streak it in this region of the plate, and then put the antibiotic disc down, and then put the plate in the incubator upside down, and then grow it for at least a day, and then pull it out and take a look at it. And there you can see the bacteria growth. And there's the disc. And this has a zone of inhibition around it. If the zone of inhibition is greater than a certain size, it is positive for the bacitracin sensitivity test. And group A, Streptococcus, meaning Streptococcus pyogenes, is positive in the bacitracin test. All other members of the Streptococcus family, as well as Enterococcus, are negative in the bacitracin test. And to be negative, you either have no zone of inhibition or a small zone of inhibition, which is less than a certain size. All right, any question about that? So those are the two tests you need to show that the beta hemolytic growth on the blood agar is Streptococcus pyogenes. Let me state that the catalase negative says that it's either Streptococcus or Enterococcus, and then the bacitracin positive says it has to be Streptococcus pyogenes. Another organism that can be obtained from a throat culture is Haemophilus influenzae. This is a gram-negative coccobacillus. Haemophilus influenzae is one of the common causes of ear, eye, and sinus infection in babies and young children. Not so common in older children or in adults. Haemophilus influenzae is a fastidious organism, but it will not grow, at least alone, on a blood agar plate. The reason being is that Streptococcus pyogenes can grow because it lyses the blood cells, and when you lyse the blood, you release the chemicals in the blood that the Streptococcus pyogenes requires. And you don't need to know this, but it's actually the factor X and the factor V that Streptococcus pyogenes gets from the blood. Haemophilus influenzae does require the V factor and the X factor. The V factor is NAD and the X factor is Hemin. And Haemophilus influenzae cannot grow on the blood agar plate because Haemophilus influenzae does not lyse the red blood cells, and that does not release the V factor and the X factor. But you can get Haemophilus influenzae to grow on chocolate agar because chocolate agar is the same as blood agar, except that we add the blood when the agar first comes out of the autoclave. And the agar is so hot that it lyses the red blood cells, changing the color from reddish to brownish. And that's why it's called chocolate agar, because this color reminded someone of, I guess, milk chocolate. And so if you swab Haemophilus influenzae on chocolate agar, you can get it growing on the plate because the blood has been lysed, releasing the factors that Haemophilus influenzae requires in order to grow. It is possible to get Haemophilus influenzae to grow on a blood agar plate, but only if you streak Staphylococcus aureus on the plate first. What you do is you get a blood agar plate and then you streak Staphylococcus aureus down this way on a single streak. Let me blow it up even further. And then you sterilize your loop and get your Haemophilus influenzae and then streak it perpendicular to the Staphylococcus aureus. And we're streaking at least all of this region. I can see the streak here. I don't know how far they went here, if they stopped here or if they went all the way there, I don't know. but at least they streak from here to there. And Haemophilus influenzae will not grow in this region of the blood agar plate because it doesn't have the factors that it needs from the blood. It will grow, however, in this region of the plate, any region near the Staphylococcus aureus. The reason being is Staphylococcus aureus, which you can kind of see the streak right there in the center of this, that's the Staphylococcus aureus growing. And then the clear region is the beta hemolysis. So most of this streak is actually beta hemolysis where the blood has been lysed. And then the X and the V factor can diffuse out of the lysed blood here up until about there on the plate. And then Haemophilus influenzae can grow in this region of the plate. Any question about any of that? Showing you the blood agar is supplying the nutrients that Haemophilus influenzae needs. It's just that the Haemophilus influenzae cannot get it from the blood cell unless the blood cells are lysed. And Staphylococcus aureus is lysing the blood cells. All right, any question about throat cultures? If there's no questions, let's move to urine cultures. Urine cultures are used to diagnose urinary tract infections. Bacteria can travel to the bladder through the urethra. It says here that the urine in the bladder is normally sterile. As you recall, at least in female bladders, they do have the normal microbiota in their urine inside the bladder, but this is normal microbiota. It does not cause a urinary tract infection. And in fact, only recently, within the last 15 years, have, I don't know, scientists and doctors even known that there is bacteria in the urethra. I mean, not the urethra, in the bladder, at least in females. I don't know about males because I've never found anyone who ever did any research looking for bacteria in the urine in the bladder in males. I don't know why, but as far as I know, no one's done the research, or at least they've not published it. Anyways, females are more likely to get a urinary tract infection because they have a shorter urethra than males. So if a pathogen gets on the urethra, it's more likely to work its way up the urethra in a female and then infect the bladder than it will in a male. Any question about any of that? All right. So to get a urine specimen, the patient or a doctor generally cleans the genital area prior to collection. And then we often use a clean catch midstream method for obtaining the urine. That's where the patient starts voiding in it where the genitals have already been cleaned. And they do not collect at the start of the urine stream. They do not collect at the end of the urine stream. They only collect in the middle of the urine stream. And that reduces the cultivation of transient organisms in the urea that come from the urethra. If you don't know, the urethra is colonized by microbes. An even better way is to catheterize the patient and stick a catheter up the urethra all the way to the bladder. The trouble with that is the urethra has microbes on it, and when you stick the catheter up the urethra, you can take bacteria and bring them into the bladder and then obtain them when you take your sample from the bladder, your urine sample from the bladder. So the catheter is cleaner, but it is not, I don't know, clean. Isn't totally clean. An even better way is to do the suprapubic aspiration, where you stick a needle through the patient's abdomen and then through the bladder, and then sample the urine directly from the bladder. Obviously, you sterilize the abdomen first with an alcohol swab, and then you're unlikely to get bacteria contamination of the urine in the bladder. However, patients do not like a suprapubic aspiration. To tell you the truth, they don't really like the catheter, and that's why the clean catch midstream is often done to obtain a urine sample from the patient. Plus, the patient can collect the sample themselves with that one. All right. Once the urine specimen is obtained in the lab, only 0.001 milliliter, that's 1,000th of a milliliter or 1 microliter of the urine is placed on a blood agar plate, a bat plate, using a special inoculation loop that measures exactly 1 microliter. And there we're seeing it there. There's the loop, and that's filled with urine right there. And what you do is you streak it this way and then streak it that way. And then you get your blood agar plate, something like that. You then will streak it on either a MAC or an EMB plate. And usually that plate, the streak will go this way, and they usually use the normal streak plate method for isolating colonies. So there's quadrant 1, quadrant 2, quadrant 3, and then quadrant 4. And this is on a MAC plate, but it could be EMB plate. So we always use a blood agar plate for a urine specimen, and that's to get both gram-positive and gram-negative, as well as some fastidious organisms can grow on the blood agar. But we also do a same streak from the same culture on either EMB or MAC, which select for gram-negatives, because the most common bacteria causing a UTI is a coliform. A coliform is something that grows in the gastrointestinal tract, particularly the colon, why it's called coliform, especially E. coli, which is the #1 species causing a UTI. And the coliforms and the E. coli can grow on the MAC or the eosine methylene blue. They can also grow on the blood agar, but the MAC and the eosine blue will remove all the gram positives, and there's usually several because the urethra oftentimes has gram positives on it. Actually, the urethra always has gram positives on it. It has a lot of species on it. And not most of them, but many of them are gram positives, probably most of them. The interpretation of a positive urine culture is multifaceted. Factors that need to be considered are the type of collection. For example, the the clean catch midstream method contains more contaminating microbes from the urethra than the catheter method, and then the suprapubic aspiration usually doesn't have any contaminating bacteria in it. The gender of the patient is important too. Females, if you're using the clean catch midstream method, tend to have more bacteria, more, I don't know, contaminating bacteria from the urethra than males. The number of colonies per mil of urine is important for the consideration. And then the presence of polymicrobial growth. If you get multiple organisms isolated, it may indicate poor collection technique. All right, once you get the bacteria growing on the blood agar or on the blood agar and a BAP and McConkey's agar, you then run some tests to determine which species of bacteria is causing the UTI. One of the tests we run is a coagulase test. The enzyme coagulase is an enzyme produced by Staphylococcus aureus, and only Staphylococcus aureus makes the enzyme coagulase in its family. The other members of the family do not make the enzyme coagulase. Coagulase is an enzyme which can contribute to the pathogenicity of Staphylococcus aureus by converting blood plasma, the protein fibrinogen found in the plasma, into fibrin, which results in a clot. So if you have the enzyme coagulase around, you'll get a blood clot, and the blood clot can protect the bacteria from phagocytosis, as well as help protect the bacteria from other factors of the host immune system. Phagocytosis happens by the white blood cells, which go out and eat things invading the body, such as the bacteria. Staphylococcus aureus makes this enzyme so that when it first infects the patient and the bacteria numbers are small, the blood clot will protect the bacteria cells until the bacteria become greater in number. And actually, you don't need to know this, but Staphylococcus aureus would then dissolve the clot because there's enough bacteria to be able to resist the host. And then the bacteria wants to dissolve the clot so it can spread elsewhere. And it can't do that while it's inside the clot. As well as inside the clot, the bacteria may start running out of resources. The important thing is to note that just that the clot can help protect Staphylococcus aureus from the host immune system, and only Staphylococcus aureus in the family Staphylococcus has the enzyme coagulase. So if you happen to know that the urinary tract infection is caused by Staphylococcus, you can run the coagulase test, and if it's coagulase positive, then it's Staphylococcus aureus. Any question about any of that? There are two tests we have for running the coagulase test. There's the slide test, where you put plasma and the bacteria on the slide, and if the plasma stays plasma, meaning no clot, it looks like that. And if the plasma begins to clot, it'll look like that. You'll see clumping in what used to be the plasma. So the bacteria have the enzyme coagulase causing the clumping like that. There's also the tube test, and the plasma is a liquid, so the coagulase negative organism will not form a clot, and you can tilt the tube and it'll run because it's a liquid. And if you tilt the tube that's coagulase positive, meaning Staphylococcus aureus is the bacteria in this tube, then you'll get it clotting and you'll see it clumping like that. All right, any question about that? So the most common bacteria isolated for a UTI is E. coli. E. coli is of the family Enterobacteriaceae. The family is gram-negative bacilli that ferment glucose, and they're oxidase negative. So we could use the oxidase test to separate out the family Enterobacteriaceae from the Pseudomonas family, because all of the members of the Pseudomonas family are oxidase positive. You could also, instead of doing the oxidase test, you don't need to know this for this lab, but you could look at the glucose fermenting, because all the members of the Pseudomonaceae family do not ferment glucose, and all the members of the Enterobacteriaceae family do ferment the sugar glucose. And then all the members of the Enterobacteriaceae family are facultative anaerobes, and all the members of the Pseudomonaceae family are aerobes. So you could use either one of these three tests to distinguish the family Pseudomonaceae from the family Enterobacteriaceae. But oftentimes they do the oxidase test, probably because it's quicker, easier to do than the other two tests. The oxidase test is looking to see if the bacteria contain the enzyme cytochrome C oxidase, which is found in the electron transport chain of an aerobe. If the organism produces oxidase or the enzyme cytochrome C oxidase, it will be oxidase positive and we'll see a blue or indigo color, meaning purplish color, in the oxidase test within 30 seconds. If you see the color after 30 seconds or you never see the color, then it is oxidase negative. Once again, the Pseudomonas aeruginosa, which is a human pathogen, as well as all the family of Pseudomonaceae, are oxidase positive. And all the members of the family Enterobacteriaceae are oxidase negative, in which case we will not see any blue or indigo color here. So it'll be something like this region here is what it would look like. All right, any question about the oxidase test? All right, so that's it for the urine and throat cultures, discussing the major tests for each culture, and then the media we use to grow a culture obtained from either the urine or the throat test. You're not physically going to be performing any of these activities, but you will be held responsible for knowing about them and how to perform them. We have some videos for you to watch on the catalase test, a real short video, and then a video on the coagulase test, and then a video on the oxidase test. Any questions about any of that? There is no virtual lab, because the only virtual lab we have runs on Flash and it doesn't work anymore. All right, let me go to the worksheet. So for questions 1 and 3, the questions are asking about the enzymes catalase, coagulase, and oxidase. For all of these questions, just fill in the answer as provided in the worksheet, meaning the lab module. If you get an answer from the AI, I'm going to warn you it may be marked wrong because the AI can find an example. Like I once had a student show that there was one case where one patient came down with a disease caused by one species, and I had to allow it because it was a real case, but I've decided in this lab and all future labs, I'm going to require the answer to come from the lab module. If you get it from the AI, I'm just warning you the answer could be wrong. And let me see, maybe this one here. And then there's one other question, which, oh, it's question number 8. I'll talk about it when I come to question 8. You can get the answer from the AI, but it may be marked wrong. Okay. So for question 1, which enzyme converts fibrinogen into fibrin? And then that's 1A. For 1B, name a microbe that tests positive for this enzyme. For question 2A, which enzyme detects the presence of cytochrome C oxidase in the electron transport system? And then for 2B, name 2 microbes that test negative for this enzyme. You can either give a species or the family name here. For 3A, which enzyme hydrolyzes hydrogen peroxide into the non-toxic products water and oxygen? And then for 3B, name 2 microbes that test negative for this enzyme. For question 4, also referring to these 3 enzymes, which enzyme is used to differentiate between the following: Staphylococcus and Streptococcus? Because obviously you could look at them under grow the cultures in liquid culture and then do a wet mount and then determine is it Staphylococcus or Streptococcus. But you can also do it from an enzyme. So which enzyme? And then 4B, how do you distinguish between Staphylococcus aureus from other Staphylococcus species? Which enzyme? And then for 4C, how do you distinguish Enterobacteriaceae, such as E. coli, from the Pseudomonaceae? Which enzyme? And then 5, concerning specimen collection for a routine throat culture, what areas of the throat should be swabbed? Give that answer there. And then what areas of the mouth should be avoided? And then 6, what is the most common species causing bacterial throat infections? It has to be an infection caused by a bacteria, and it has to be a species name. 7, what test is used by microbiologists to differentiate between beta hemolytic group A Streptococcus and other beta hemolytic Streptococcus species isolated in throat cultures? So we already know it's beta hemolytic, and we also already know that it's a Streptococcus species. So we only need one test to then show that which species it is. And what's the test? And then 8, answer the following concerning a child's throat culture. 8A, state 2 non-selective agar plates that are inoculated from a culture obtained from the throat. And then 8B, what organisms can be isolated on CAP, but you cannot isolate them on BAP? CAP being chocolate agar, BAP being blood agar. 9, list 2 media that are used to grow urine cultures. 10, list 3 reasons why urine specimens should be rejected for culture. You can see the appendix for that. Oh, I forgot. 8A, 8A is a question that you need to get from the lab module. If you get it from your AI, it may be wrong. If you do get it from AI and you happen to state what the additive is you need to add to the media, I think I will allow that. But most of you will just say a media, and that won't work. The AI is either wrong or they're not realizing that someone is adding an additive to that media. Let's see, we've talked about 9 already. 10, list 3 reasons why urine specimens should be rejected for culture. See the appendix for that. And then 11, how much urine is placed on the surface of a blood agar plate? State that there. 12, what is the most common bacteria isolated from patients with bladder infections? You should mention a species there. And then 13, identify the Gram reaction, meaning what's the Gram stain, and the cell morphology. What is the shape? Do not give me the growth arrangement of these bacteria. So Pseudomonas aeruginosa, give me the gram stain and the shape. E. coli, the same. Staphylococcus aureus, Streptococcus pyogenes, and Haemophilus influenzae. Any question about any of these questions or anything about the lab? All right, go ahead and get to it. I'll be here until the last student logs out or 8 o'clock, and I'll guarantee you it'll be when the last student logs out.